In this lecture, I will discuss the various steps or what are the various aspects one has to consider for developing any algorithm. So, last class we discussed about the scripting language, that is pattern action statement, that is awk programming. So, what is awk programming? Script, the data driven language, right. So, this is a lot of potential applications, mainly when you handle data sets, or you can manipulate the outputs given from any of these programs, right, mainly for this massive data if you want to handle. So, there are various options available in awk programming. Either you can use one line commands, or you can make a kind of program, right, to run in Unix environment, right, to manipulate several data. Also specifically we discussed about the usage of these awk commands to manipulate PDB files. For example, how to get the C alpha atoms or how to extract the sequence information, right. So, also the how to get the data for the atom records and so on. Then also we discussed a little bit about the calculations we can also use for various arithmetic operations, right, also make proper outputs, right. You can use this awk in an efficient way. So, now the question is, okay, we discussed about various aspects of bioinformatics and databases and the tools, right. So, if you want to develop your own algorithm on different aspects, what are the various aspects we need to consider, and what are the available problems now you can tackle, right, in bioinformatics, right. I will explain now. So, first we need to identify research problem. So, we discussed various types. The two major types of research problems, one is a classification problem, that means yes or no, right, either yes or no. So, various aspects, whether the particular residue can bind or no, right, this is stabilized or not, right. This is a kind of classification problem. And the second one is a kind of exact real values, right, for example, is 4.5, right. So, what is the temperature? This is 21.2, right, exactly you can tell, right, this is the actual value. So, two types of problems, right, this is generally occurring in various fields of bioinformatics, right. So, I will explain some of the aspects in this lecture. So, once you define or once you identify your research problem, then how to proceed, right. There are various aspects we need to consider when you develop any algorithm. First, I will for example, the data set, that is very important, because your for the final results and the model mainly depends on your data set. Earlier we discussed about the non-redundancy, right, how to construct non-redundant data sets and what is the importance of non-redundant data set, right. I will explain the details, right, about all these features. Then once you make a data set, then we need to identify the important parameters or important properties, what we can learn from this data set. Anything is very specific. For example, if you consider the classification problems, for example, A or B, so what is peculiar or what is very important in A and what is very special in B. So, we could extract this information, then you can use this information to classify these two groups A and B. So, then we use some methods, there are various methods like statistical methods or the machine learning techniques, right. You can use this subjectively, you can see put some conditions and you can classify, okay, if this particular value is positive or negative, then this is class A or class B and so on. Or you can use some machine learning techniques, here this you can the machines, if you give all the input data and the output data and your features, the machine will train the data and will give you the most probable output, right. Then you can use that model for predicting any new set of data. So, once we get these methods and get the result, then we need to validate. First we need to assess the performance. Once the performance is good, then you need to validate whether this is valid for different sets of data, right, that I will explain. And the applications, what are the potential applications for your method and what are the various aspects you need to consider to get more visibility of your particular program. So, I will tell you some of these research themes, right, I just now discussed. There are two different types of problems, one is a classification problem, another one is exactly predicting the real value, right. Classification problem I will tell you some of the important areas. One is whether you can predict the secondary structures, right. So, we discussed about secondary structures. What are various secondary structures? Helix, strand, coil, turn, right, whether can we predict. Then we discussed about the DNA binding proteins or the RNA binding proteins, whether you can identify the sites, okay, this is binding, this is not. Likewise, you can see the membrane. So, where the residues are located, right. So, then you can see some functions, okay. So, if you have a sequence, whether this can perform the transport or not, right, you can do that. Then we can see the mutations, that also we discussed in the earlier classes, whether this will stabilize or destabilize. I will tell you more details. Okay. So, if we take the secondary structure, so if you see some region they are helix, some region is strand, right, and also different types, right. So, if you have the sequence and if you do not know about these secondary structures, we can predict whether in particular segment, right, this can form helix or it can beta strand or it is a coil. If you want to have a two state problem, you can see this form helix or not, or strand or not, right. That way you can see classification problems. You can make two state classification, or you can say three state classifications, or you can see go for the multi state classifications. So, in this case, depending upon your secondary structures, either you make two states or you can make three state. If you want a three state, you can take helix, strand and coil. Two state means you can set helix or not, or strand or not, right, or you can have the four state classification, helix or strand or coil or the turn, right, you can do that. Then we go with the accessibility, right. So, how to get the accessibility?
By probing
1.4 angstrom radius water. So, you can get the residues which are accessible to solvent, that you can get the data. So, there are various programs to get the accessibility. What are various programs to get the accessibility? DSSP you can get, access, NXS, many programs you will get the accessibility. Likewise, the secondary structure, what are various programs we use to get the secondary structure?
Again DSSP.
Right, you can see DSSP, right, get the secondary structure assignment. Also, when the hydrogen bonding pattern you can see experimental data. So, now you have the solvent accessibility. This is the protein. So, you can see this is the protein. So, you can use the concept of solvent accessibility and finally, you get this figure, right. So, here there are two different aspects. One aspect is you can see the residues which are in buried, right, which are residues are in buried, especially you can see these residues, they are in buried. If we have any particular sequence, for example, this is a sequence. So, take this one, this is buried or exposed. So, this is buried or exposed, you can check the two state classification. You can make any cutoff and you can develop model. So, to see in this protein these residues are buried and which residues are exposed, two state classification, or you can make three different classes. You can take buried, you can take exposed, and you can make a cutoff that is partially buried or partially exposed, right, you can see that. So, you can make any cutoff. For example, if you take the buried, you can take a cutoff of 5% accessibility, right, and exposed you can put more than 50%, and in between you can put partially exposed, right. If you take two state classification, you can put a cutoff. For example, it is 20%, less than 20% take a buried and more than 20% you can take it as exposed, right. Likewise, for any amino acid sequence, you can classify these residues based on solvent accessibility. Why do you need the solvent accessibility? Once you predict the accessibility, then you can see the location. So, which residues are interior, which residues are at the surface, and which type of interactions these residues can make. So, if you have a sequence, you can predict, right. There is various methods to predict the accessibility, right. You can also develop a model with better accuracy, right. So, if you have the sequence, you can use some parameters and you can predict this two state problem. So, now for example, this is the kind of membrane proteins. So, for example, if you are interested in this type of membrane protein, what is this type of membrane proteins? Alpha helical proteins, right. This is the transmembrane helical proteins, this is strand proteins. If you are interested in this one, so if we have a set of sequences or if you have two sequences, you can say this sequence belongs to the strand type or not, or this protein belongs to the helical type or not, right, because these proteins perform different functions, right. So, in this case it is very important to identify the proteins in any genome, right, either this is helix or this is strand, right, you can do that, or you can say this is membrane protein or not. If you take this as whole, right, these are membrane proteins. Here if you see this is a globular protein. So, if you see you can get a sequence, you can identify whether it is a globular protein or it is a membrane protein, right, you can see that. So, right. So, now if the case it is a protein is a membrane protein, so you identify a protein as membrane protein, right. What is a membrane protein? The proteins which are embedded in the biologic membrane. So, if we have this is a membrane, this you can see this is a membrane, right. So, the protein there are some several residues which are embedded into membranes. So, the question is if you take one particular residue this inside the membrane or not. So, take another residue here this is in the membrane or outside the membrane, right, because this will have functional implications, right, because they to understand the functions, right. So, it is very important to see whether any residue is located within the membrane or in the surface, right, this kind of classification problem. So, go one step further again. So, here we are talking about the binding. Till now you discussed about the single features, right, based on the structure either the helix or strand accessible or not accessible or the proteins membrane or not, right. So, now we getting a little bit complicated. So, we have the composition. For example, if you have DNA binding proteins, right. So, you have protein here, right, and you have the DNA, right. So, you can see some part, right, you can see this interacting, right. So, protein has one sequence and DNA has a sequence, and if we see the sequence the question is whether it is binding or not. Then we can see it as a classification problem, right, whether it is where it can bind. Also you can see whether a particular protein is binding the DNA or not. For example, if you see different types of proteins, this is green one is DNA binding and the blue one is the non binding. Then if you take any protein, right, whether this belongs to this group or this belongs to this group, is binding with the DNA or not. If it binds the DNA, then the question is any residues that residue binds or not. For example, this residue is A, this residue is R, right, which one binds? R binds, right, this is binding, this non binding. So, we can do that. So, likewise if you see the DNA you can see protein complexes, protein RNA complexes and protein ligand complexes, right, because these are very important in the because they perform various functions we discussed in the previous classes and structure based drug design, right, they are important to identify the binding sites and the pockets, right. So, in this case if you have a sequence you can develop methods to identify the residues which are binding, right, this is a classification problem. So, this is another important one. So, there is some cases if you have a protein, some regions are missing in the structures. For example, here some these residues in red. So, they are missing in the protein side, but when it makes a complex with other molecules like other protein or the DNA or the RNA, in this case they get the structure. So, the partner one they promote the folding, right. So, in this case they get the structures. So, if you give a sequence, right, you can predict which regions are disorder regions from the sequence. If you know the structure we can find okay here the density map is missing. So, your structure is missing. So, this is a disorder region, but if you have the sequence is it possible to predict whether this residue is disordered or not, right, that we can do that. For example, here these are regions, right, these are the ones we have the structure in the complex, but this is missing in the case of these free proteins. Then the next aspect is you can see got a stability. We discussed about stability, right. For example, if you mutate a particular residue here, this residue is in contact with various other residues, these are the residues which are in contact. If we mutate this residue, either it may establish more contacts or it may break some of the contacts, this may increase the stability or decrease the stability. So, for example, we discussed earlier alanine to valine, depending upon the place where we mutate, this can either well packed to increase the stability or it can have the steric and decrease the stability. Like the alanine to aspartic acid, either a surface you can make the electrostatic interactions with lysine and arginine, or if it is in the buried then this will disturb the protein, destabilize the protein, right. So, likewise if you have any mutation, for example, alanine 15 is mutated to valine in a particular protein, whether it is stabilized or not, The question is okay, two state problem, this will destabilize or destabilize, right. So, you can take this as a problem. So, like you have the several diseases. For example, glutamic acid 6 to valine, sickle cell anemia and hemoglobin, take P53, there are many mutations, right, which cause cancer. So, here also you can see whether this mutation, right, it is the causing tumor or not, right. This is a kind of two state problem. So, here let us show one example. This is the data for the epidermal growth factor receptor, right. There are two different domains, and if you see some letters in green, some of them in red, and the green one are the drivers, that means they cause cancer, right, for the progression of the cancer. So, the green ones, right, this is the passenger, that means even if you have the mutations, there is no change. So, there are several types of mutations, the red one is the driver mutation and the green one is the passenger mutation, right, you can see. Now, if you have a particular mutation, for example, is isolation 706, ILU 706, if you mutate to other residue, for example, arginine, what will happen? This is a passenger mutation or a driver mutation, right, you can classify, you can take it as a classification problem, whether driver or it is passenger. Likewise, there are several image processing, that you can do the images, you can scan several images. Here I show two types of images, right. So, here one is this benign, there is nothing to do with the cancer, but here is a malignant, right. So, can we see these images and then predict whether this is cancer prone or this is neutral one, right. This will mainly deal with the skin cancer. So, you have the two types of data, either you have the numerical data, right, or you can have the image data. So, even both cases you can have different types of problems, like you can classify into different groups. Likewise, you can see the several problems in bioinformatics that you can classify, right, these are there. Okay, then you go with the real value prediction, right. Here there are various problems, right, which can give exact real value. For example, stability change upon mutation. For example, A15 is mutated to valine. Earlier we discussed whether this will stabilize or destabilize. This is a classification problem, but you can see also this will decrease the stability of 1.2 kilocalorie per mole. So, exactly real value. So, if you mutate in a specific residue, you can measure or you can predict the exact value, okay, this will change the stability by 1.2 kilocalorie per mole, this is a real value, you can do that. Like binding affinity of protein complexes, if you have one protein, this another protein, if you combine these proteins, right, when you form the complex, right, the affinity, what is affinity, how strong these two proteins can interact. Like protein folding rate, we discussed the folding rate, how fast or how slow a protein can fold, right. So, you can see whether if we have a protein sequence, can we predict this is the folding rate, right, that you can predict. Then solvent accessibility, right, earlier we discussed two type of classification, right, the buried or exposed, right. For example, if you have ASA value of 12 angstrom square, so we take a cutoff of 5 angstrom square, then this is predicted as exposed, right, predicted exposed, right, with different cutoff. If you put a 20 angstrom square cutoff, then this is predicted as buried, right. So, now you can exactly predict the values, okay, this value is 12 angstrom square, you can predict, okay, this accessibility of particular residue is 12 angstrom square or 15 angstrom square, exactly you can predict real values. So, now we discussed about the different types of classification problems, right, some of them I discussed, there are many problems available in the literature, right, for the applications to the bioinformatics. So, now the question is, for example, if you identify your problem, you want to classify, for example, driver mutation and passenger mutation or the stabilizing mutants and destabilizing mutants, what you have to do first? It is very important to construct data set, because data set is very important, because we need the experimental data for developing any model. If you use any predicted data, that is also predicted, so then you will give the introduce more bias, right. In this case your sensitivity or your reliability will decrease, right. So, you need the reliable data. So, in this case we discussed about various databases. What are various databases we discussed earlier?
Protein sequence database,
Protein sequence, for example, UniProt,
protein structure, PDB,
structures PDB.
DNA sequence,
Okay, for example, the DNA sequences. What are the database for DNA sequences? DDBJ,
GenBank,
EMBL, GenBank, and protein stability, Protein database, right, enzymes, Blender database. So, several databases are available in the literature for the bioinformatics, reliable databases, right, widely used databases. Fine. First, if you want to develop any method, it is very important to construct a reliable data set, because all your results, right, depend on your data set, because data set is very sensitive, right, as much diversity you can add, right, and you have to get the as much reliable data as possible in the data set. So, you can, for example, if you are interested in the stability, you can go to the Protein database and you can get this data, right. When you get the data, if the full set of data we get, right, then we need to reduce the redundancy. Redundancy reduction is very important, otherwise what will happen if you do not reduce the redundancy, what will happen?
results will be biased.
We introduce bias. So, in this case model is biased with that particular data, because if you only have more data in same size, right. So, that will be biased with that particular data to improve the performance, right. Finally, if you have new data, right, then the method will fail. In this case, it is very important to have the non-redundant data set. How to derive the non-redundant data sets? Yeah, for example, if you have sequences, then you can use the steady heat or Pisces or blast class to reduce the redundancy with various cutoff values. If you have the mutations, right, you can get the unique mutations, right. For example, A to A or 15 is mutated to valine, for example, 5 data are available, and you can consider the unique ones, right, for setting the analysis. Otherwise, if there is plus, your results will bias to this plus. So, you can make the non-redundant data set. So, if you have this is the main data set and you can get the non-redundant data, this is the non-redundant data set. So, this is the redundancy we removed, right. So, we have the non-redundant data set. Now, the next question is, when you develop a model, if you use all the data, then you can, the models will fit with respect to all your data. In this case, we do not know the reliability for the new data, right. In this case, you classify this into at least 2-3 groups. So, you can take it as a main data set, this you can use it for deriving your features or trying to assess the performance of your method, right, within your data set, whether your method can fit the data, right, that what you have. The method is not able to fit, then you need to find new features which can fit your data, right. So, then you keep new data sets, for example, to test or completely blind. Now, you are not taking any information from this data, right. Sometimes you take some of the information to derive the features and you can use leave some other information, they use as a test set, and some cases you are not at all using any of the data to in your data set or derive any features, right. For example, if you calculate the propensity, you can get the propensity with the 100 proteins, then you can use some proteins, right, to train, and some proteins you can use test, and some cases completely new proteins you do not take any information from that also you can keep. In this case, in case there is no information you consider from that, right, that you can use as a blind data, completely new data, right. Likewise, you have the different data sets, one is the main data sets that you can train and cross validate, right, and you have the test set, right, and also you can use the blind data that is completely new. For example, if you take a new data, right, how to take this in the, if you take any classification problems, you can leave the new data, but take the old data for the data sets, because even if you get some features, right, for example, if you take the heterogeneity values, you take from the literature 20 residues, but we do not know which proteins they consider to derive the values, right. If you use this test set, even your data set is new, if you use a heterogeneity as a feature, then the values that are included in this test, but with a completely new data, so that means this data is totally new, no information is taken from that particular data, right. So, this is how we take it as a blind data set, fine. So, why do we need 3 different types of data sets? to check reliability, right, because otherwise if you check with this data set you can get the accuracy of 97% for example, you will be very happy. So, 97 is a good number, right. So, then you see think that okay your method is working fine, right, because we do not consider that there is some overfitting in the data. So, here your method is try to accommodate all the your data with some specific features. So, you do not know whether this can also work with this new data, that is the one we need to predict, right. Otherwise, if you do with the same data set it is a kind of predicting yesterday's weather, right, but we need to predict tomorrow's weather. So, in this case we need completely blind. So, we do not know, right, with the present conditions, right, and the other environmental factors we will think okay tomorrow may rain, right. Likewise, we have to get the information from this main data set, and with the available conditions with the known information you can predict the blind data, right. This is why we need more data sets. It is very sensitive because data set is very important. So, even if you take time to derive data set, so you have to be careful to make a good data set. So, this is the reason if you make a good data set you will get a lot of citations, because people do not want to do that, because it is very time consuming, right, compared with the other steps in developing algorithm. So, they usually they do not want to do the develop data set, they try to find any data set is available readily available, right. So, they get the readily available data sets and they train and they use their own methods on the data sets, right. So, if you make your own data sets reliable data sets, then that will be used by many researchers in the literature. So, now when you have the data set, okay, so you have the data sets for the training or the whatever the blind or the data sets, then what can we do next? So, we try to understand and see whether any intrinsic properties within your data set, can we identify any important features which can fit your data, right. For example, if you have the two proteins, one is a globular protein, one is a membrane protein, right, can you find any information, for example, with respect to the amino acids, any specific residues, right, are biased, right, are over represented in any of these proteins or under represented in these proteins. Then if you see new ones you can say okay this residue is over represented here, for your test set also it is over represented. So, this could be used for identifying the type of protein. So, we have to find the different features, right, because the various features we discussed. What are various sequence based features?
Amino acid composition,
Occurrence, composition,
average amino acid property,
average properties, molecular weight,
HLP,
right, profiles, conservation score. So, we can derive various properties using the sequences. Like if you have structures, there also you can derive various features, for example, accessibility,
second
contacts,
contact order,
potentials, right. So, you can see derive various surrounding hydrophobicity, right, we can derive various features using the structures, right, contact order, long range order and so on, right. So, now if you have a sequence or if you have a structure, you can think of the reliable features which can address your problem, right. For example, if you are interested in the binding affinity of protein complexes, right, this is like high affinity or low affinity. So, relate the features which are relevant to the affinity. For example, if you take the affinity we discussed of protein complexes, so which interactions are important? Electrostatic and the cation pi and the aromatic interactions. So, in this case you can look for the residues which are responsible for this type of interactions. And the second one, instead of looking for the whole sequence, if you are able to identify these binding sites, whether you can see any dominance of these residues at the binding interface, right. So, if you do it properly with the features which are relevant to the particular problem, then you can perform better, right, in your prediction accuracy. So, you mentioned some of these features like the physicochemical properties or specific interactions. For example, what are the physicochemical properties? For example, molecular weight, you can just shape,
charge,
okay, charge, hydrophobicity and so on. What are the interactions? You can electrostatic interactions, van der Waals interactions, hydrophobic interactions, so on. So, you can see the composition that we discussed earlier, pair preference, you can see in specific motifs, right. For example, there is a motif G, X, 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 G motif for membrane proteins, right. For GPCR we have this DRY motif. Also we discussed about the beta barrel membrane proteins, right, that is glycine, then polar, right, and hydrophobic, polar, hydrophobic, this motif, right. The various motifs, right, are present for different types of proteins with specific functions or specific structures. So, you can derive these features, right, or relevant features, right, with your sequence.